One of the goals that Genes Conference has is to be able to bring physicians and embryologists to the same table and create an environment where we're hearing both parties of the IVF, field. Typically you see one conference just for embryologists and one just for physicians. Now we're bridging through that gap. Today we're going to be talking about current status and future of reproductive genetics. And I'm going to try to be a little bit brief because I have seen wonderful presentations that are similar in some ways to what I have been, what I have prepared. So this is the classical picture of where we were, a few decades, and then with the fish technology, and then we went to microarray technology, which was quite an improvement in the scope of technology, sensitivity, specificity, etc. And then in 2015, 16 is where NGS took place. So when we look at current chemistries used for PGTA, and I don't know how to use the pointer, hopefully that's the pointer. Okay, I'm gonna escape the pointer. So for whole genome amplification, so you have this random amplification that every lab uses these days. It's a relatively low resolution, but it gives you a whole scope of what happens in the chromosomes, right? If you have an extra copy, missing copy, then there is a chemistry that is more targeted, where you look at specific SNPs, specific spots in the genome, and that gives you also some sort of view, but the whole random genome gives you a little bit a bigger picture. Santi will be talking about whole genome sequencing base per base, where you actually look at maybe 80% of the genome or higher. So when we look at sequencing platform currently used across the industry, so you have Illumina platform, Thermo Fisher platform, some of the labs will be using SNP array. And then what we have not used so far is PacBio, which is a chemistry that gives you long reads, about 1,000 base pairs. So compare 1,000 to approximately 150, so about 8 times more. Illumina gives you about 36 base pairs, so even shorter. But with nanopore, you can sequence 100,000 base pairs. So imagine the scale of sequencing a single molecule that have over 100,000. If you're testing human genome and amplified from a blood sample, you can reach millions of base pairs. So imagine what that will give you as far as translocations and inversions and genome modifications. So we have not tapped into that yet. So some of the limitation we see is the resolution, right? Resolution is 5 to 10 million base. There are certain platform microarray that gives you a little bit better. We have worked on triploidies. Triploidies is less than 1% of human embryos. XXY and XYY are detectable. The algorithms don't pick them up, but they are detectable. You can adjust the algorithm to make that call. The 69XXX are hard to detect because it's like a 3-copy genome. It's exactly like a 46XX, but we have found ways of clustering the data and looking at more creative ways of mapping the data to be able to distinguish that karyotype. And then, as you guys know, in the course of IVF, there are clinics that wanted to know the origin of the embryo, whether if it belongs to the couple or it doesn't belong to the couple. It's a big decision for a clinic to transfer an embryo. With SNPs and with STR, STR stands for single tandem repeats. Those are repeated sequences happen in the genome that are used for paternity testing. So if you take 2 individuals in this room, they will have, and you look at one STR marker, they will have 2 different repeat number. So using those tools, you can actually identify whether the embryo belongs to the couple that you are transferring the embryo to. And you can also solve problems like maternal contamination, etc. So next point is invasive versus non-invasive PGTA, which I will talk about in a few moments. And last one is, if you have the ability to do whole genome, of course you can look at a lot of things, inversions, translocation, gene copy number, etc. So mosaicism has been, addressed in many papers. Dr. Swain just made a very nice presentation on mosaicism. I'm going to approach it slightly from a different angle. So without giving you the introduction, you know what mosaicism is. When you look at sequencing platform, there are differences. And the way we know that, we look at labs that are using NGS. And just by looking at the rate of mosaicism, I can tell if they are using Illumina or they're using Thermo Fisher. And the answer to that is a little bit more complicated, but I can go into it. So we have done some modification to reduce the mosaicism rates by increasing the size of the read, of the sequencing read. Because at the end, the noise, and I can speak about this in a moment, the mosaicism has these artifact components, which is the majority of mosaic calls that we see, they're artifacts. And artifacts comes from a variety of places. They can come from the sequencing platform you're using because they are shorter read, they don't map to the genome the way longer reads do, and that gives you some noise that transcribed into mosaicism. We have seen mosaicism between labs, within the same lab, and so it spans from the genetic lab all the way to the IVF lab and the technology that is being used for that purpose. Here we're looking at 145 embryologists that use our platform. You can see mosaicism rates ranging from 0.5 to 5%. So, obviously, like Dr. Swain was saying, you can compare two different labs. They may have two different culture systems, some of the labs are more controlled than others. You'll see differences. But we have also seen differences within the same lab between two embryologists, a difference of 3 to 4 times higher. And that's, tells me that the way the embryologist is doing the biopsy can also influence the mosaicism rates. If you are very conservative and you're taking a very small biopsy, then you're going to get more noise in the data, which will be interpreted as mosaic. And so we looked at a clinical outcome from mosaic embryos. This is not a published data. On the right-hand side is a published data, but on the graph is our internal data. We don't have a lot of embryos that have been transferred, so the data is small. But in blue, you see the implantation rates, clinical pregnancy, biochemical miscarriages. You look at the gray, that's mosaic transfers, and they are not too far, especially if you look at the pregnancy rate, 40 versus 48, not too far from euploid class. And bear in mind, this is 2.5% mosaicism rate, so it's not like we are giving the clinics a lot of mosaics. They are working with a very narrow low rate of mosaicism, low level of mosaicism, and you can see they're behaving almost like a euploid class. So this is a study on miscarriages, analyzing miscarriages, short-term and long-term culture. They will take a POC tissue, then they will culture it, and they ask the question. So in that study of miscarriage study, they looked at 1,000 patients. Then they take the POC tissue and they will culture it short-term and long-term. And then they ask the question, are there differences in karyotype? The differences are attributed to mosaicism, the way they define it. But when I look at Jason's presentation, those differences can be also just stressors in the culture system that you're using, right? Culture system can introduce stress in certain in certain conditions. And so the rates of mosaicism that they found here was 3.5% based on short-term and long-term culture. So that study, 1,000 patients, found 3.5%. So I think the mosaicism rate is way lower than 3%. When we get rebiopsy of mosaic calls, we get only 0.5% that are confirmed. That's what we see. So the point I'm trying to make here is Mosaicism does exist, but it's not that common. And I'm going to go into another slide. I'm sorry, I'm doing a virtual presentation here on my laptop. But in this case, we've looked at the distribution of aneuploidy from chromosome 1 to chromosome 22 and X and Y. And we asked the question, what is the frequency of each aneuploidy in each chromosome, right? So we found that chromosome 15, 16, 21, and 22 are more common. Why? I really don't know, but they're more common. There are maybe we can address this from evolutionary aspects or things like that that makes those chromosomes more prone to aneuploidy. But the reality is, when you look at miscarriages in this study, chromosome 15, 16, 21, and 22 were higher. When you look at the population of embryos, they are higher. But when you look at mosaic calls or mosaic embryos, the distribution is flat, is noisy. And what that tells me is what we call mosaics are really just noise, mainly noise. Okay, back to the presentation. And awesome. Yeah. Okay, so just a couple of slides. So this is the distribution I was talking about, that you see a random distribution in blue, and then the CVS and the embryo looks very similar, with 15, 16, 21, 22 being more common. Now, when we look at age, we don't see any impact. If mosaicism was a non-disjunction event due to errors with age, you would see more errors, right? But we don't see that. If anything, we see a slight decrease. That tells me that mosaicism is really not as common. When we look at clinical outcome, this is from 185,000 embryos that we've tested, right? These are all the cases we have collected of miscarriages, reported miscarriages, that are due to mosaic embryos. So you can see we have collected literally 6 out of these big samples. That's how uncommon it is. And the percentages are ranging from 5 to 25%, and we have a 40% case for a mosaic XO. And we have two birth, one with 25% trisomy 21 and the other one in XO. So back to the improvements. So this is just a diagram showing you how SNPs can be used to identify triploidy. So in a typical scenario, the SNPs are either mom or dad, so you will have either 100% copy number of one SNP or 50%. But when you have a triploid, you have 2 from mom, 1 from dad, and 2 from dad, 1 from mom. So you get now the 66%, 33%, and then you get the 100% and 0%. So you see additional band when you are mapping the SNPs on the genome. This is one point. In yesterday, we heard that if you don't believe in mitochondria, then you have to look at it twice. That you have to look at mitochondria, whether if something we should look at. Now, I'm going to give you my story about mitochondria. So when we look at the mitochondria, is just a way of quantifying mitochondria in embryos. When we look at the differences between euploid and aneuploid in terms of mitochondria, does it tell us whether this embryo can be euploid or aneuploid? We don't see differences. But when you look at the data from the embryo development aspects, meaning that you're looking at day 5 and day 6, and you're asking the question, okay, in day 5, give me the best score, the best mitochondrial, because mitochondria decrease over time. So if you have very little, that means the embryo is growing very fast. So if you ask the question on day 5, give me the lowest mitochondria, and on day 6, give me the highest mitochondria, now you're looking at the impact on development, right? You look at the fast developments and very slow developments, right, in embryos, and you're adding the mitochondria on top of that. When you do double selection, you can see the clinical outcome being different from day 5 to day 6, or from high mitochondria to low mitochondria. And the reason behind that is when you look at general population without double selection, you get situation with embryologists
will grow the embryo to day 6 because that's how they practice. It's not necessarily because those embryos are slow at developing. So you get a lot of mixed bags where embryos they stay 6 days just because it's a normal practice in that clinic, and it's not really because they are slow at developing. So when you force the data to be segregated in this manner, then you start seeing more differences in outcome. Non-invasive has been the promise of PGTA. We all thought this is wonderful. We don't have to do any more biopsy. You have more media to work with. We've published a paper on spent media a few years ago. The reality is when you look at papers right now, all the literature combined, the correlation is about 80%. So you can predict about 80% of the time if an embryo is euploid. About 10% of gender discrepancy, meaning that the embryo you think it's a female, but it turns out to be a male. And that is because we have quite a good amount of maternal contamination that will overpower chromosome Y. So you have lots of X's in the media that will overpower XY, and you see more of a normal female pattern rather. And so I think adding markers like STR markers and SNP markers will help us identify when you have a maternal contamination so you can rule out that sample. What I'm going to talk about in the next slide is how that spent media can be utilized for other purposes. So what we're trying to do is we're trying to look at spent media for metabolic profiling. So if you look at media itself without embryo growth, you're going to see amino acid, you're going to see serum albumin, you're going to see some of the sugar and etc. So, and you can pretty much identify each component if you want. If you run it on a high resolution mass spec, you can identify the peaks and you can tell what you have in the media. What I like about this is when I look at Jason Swain's presentation, them trying to control the oxygen and the CO2 and say, okay, stressors, whether stressors have an impact on euploidy. With these tests, you can actually look at the metabolites and look at the stressors, the actual stressors in the metabolic profiling. So you can tell, okay, how much, say you're looking at hydrogen peroxide or even oxygen or anything that you want to measure. It doesn't have to be only CO2 or oxygen. You can go beyond that. Molecules that are creating stress for the embryos. And it could be a really good way of doing a QC in a laboratory. So imagine you have a mass spec access. You can tell if your lab is creating an environment that is very stressful to the embryos. That's very, very powerful. Beside that, you can look at, like yesterday we were hearing about the NADH and FAD, that is TCA cycle, the glucose metabolism. And they were using images, and images give you that limitation that you only look at one thing. But with this mass spec, you can look at hormone, FSH, for example. We know that certain embryos will secrete FSH as a normal mechanism of embryo development being ready for implantation. We know that there are certain molecules that are used for invasion, for endometrial invasion. Those molecules, you can look at them in the mass spec data. This is how it looks like. On top you have the media, and then on the bottom you have the embryo growing in the media. If you try to look at this data 10 years ago, there would be no way of getting a good correlation with euploidy and implantation rates. And the reason is because in the old-fashioned way, you will look at peak one by one, try to make sense of it. But the reality is every embryo is unique. Although you may have certain signature of a good metabolism, the reality is no embryo is identical to another. And the only way to do that is you have to use artificial intelligence to look at patterns that are correlated with implantation and pregnancy rates. So that's why we're going back with metabolic profiling, but this time we're using AI to solve the scoring and the identification of good embryos and bad embryos. So polygenic disorders, I think we heard yesterday a good presentation on how to use biobank data, genomic data, Those are whole genome sequencing projects that done on hundreds of thousands of patients. How can you reclassify those patients by fertility, infertility, for example, PCOS, and you can look at high rates of miscarriages, etc. And you can regroup, you reclassify them, and then look at the genome data and find markers that correlate with certain traits. And those markers can be utilized as a molecular test to identify whether a patient need to be stimulated one way or another. So you're using genomics to tailor and customize the treatment to the patients. Full genome sequencing, I'm not going to touch about this because we have a wonderful speaker, Dr. Santiago Muñe, who's going to talk about full genome sequencing. I'm just going to say that there's an enormous amount of information you can extract out of the embryos. And obviously, artificial intelligence has reshaped the way we think and the way we look at data. And then just imagine, so what we've seen yesterday, people talk, the presentation talking about embryo selection and looking at oocytes, and some of folks are looking at sperm selection using AI. But AI goes beyond embryos. We know that embryos is not the whole story. So you have the endometrium, you have the patient side. So in an ideal world, you have an AMR system that captures every single detail of the treatment, the patient history, the age, BMI, the stimulation protocol, the level of FSH and LH. And if you can use that information to train the algorithms based on thousands and thousands of patients, you may be able to get a real-time support for your practice. You may adjust the dose in real time. You may, change the protocol. And that's the promise of the AI, the way I see it. From an embryo perspective, the way it would look like in our testing, basically you will have the typical reports, but in addition you will have kinetics of the embryo, and you will have the AI score based on whatever you have designed as far as AI. And then there is a ranking system, and then you choose the embryo that is euploid, but also from an AI perspective is more correlated. So in the ideal world, to me, selecting, if we're gonna go into the non-invasive route, you have to look at genomics, metabolomics, and you have to look at the embryo images, and you have to incorporate the cycle information. It's gonna be a more of a global picture of looking at it. Sorry, I'm gonna skip. You're gonna look at all these things in combination in one picture, and then I think that's what would make next generation IVF a lot more efficient as far as outcome. Going back, so we talked about artificial intelligence, lab automation. Jose yesterday gave a wonderful presentation on lab automation. I think this is going to continue to grow as we see microfluidics, we see automation of the biopsy. I think tracking of vitrification and wireless technology, all that is going to come into play where there are less need for manual work, and embryologists will focus more on data and will focus more on looking at systems and monitoring rather than doing the work. And so we have accumulated 185,000 embryos. So we're looking at the big data, and then 120 clinics. We got about 300 physicians, more than 150 embryologists, and the goal is to try to provide the clinics with some data to use. So for example, you can compare different stimulation protocol. You can look at your embryology team and say, okay, where do I see differences in rates of mastocytism and aneuploidy and stuff like that? So an unlimited number of possibility. And with this, I was told time is up, and thank you so much. This is my team. I appreciate it. Thank you.